Imagine if you could take a single, totally invisible strand of DNA and, in just a few hours, turn it into a billion copies. Well, that's not science fiction. That's the power of a technology that completely changed the world of biology. It's a technique so powerful, people call it molecular photocopying, and it's the key to unlocking just about any secret hidden inside our genes. So think about this for a second. For decades, the huge challenge wasn't actually finding DNA. It was getting enough of it to study. A microscopic sample from a crime scene or a tiny fragment from a fossil, it just wasn't enough to do anything with. So how on earth do you solve a problem like that? Well, the answer is the polymerase chain reaction, or as you've probably heard it, PCR. At its heart, it is an incredibly clever and powerful way to amplify a specific piece of DNA you're interested in. And that molecular photocopying nickname, it's perfect. That's literally what it does. It finds the exact page you're looking for in the book of life and just hits copy over and over and over again. And this isn't some obscure, newfangled technique. It was actually invented way back in 1983 by scientists named Kary Mullis. And get this, he won the Nobel Prize for it just 10 years later. That's how fast its impact was felt. Today, you'd be hard-pressed to find a molecular biology lab anywhere on the planet that doesn't use it. It's that fundamental. So, how does this molecular photocopier actually work? Well, you can think of it like a recipe. It all starts with a few key ingredients that you mix together in a tiny tube. So let's break down what you need. All right, think of it like you're building with the Legos. First, you need the DNA template. That's your original instruction manual. Then you add things called primers, which are like little sticky notes that you place to mark the exact start and end of the section you want to copy. The real star of the show, though, is an enzyme called DNA polymerase. That's the master builder that's going to assemble all the new copies. And of course, you need the bricks themselves, the nucleotides, A, T, C, and G. The buffer just makes sure the whole environment is perfect for building. But here's the thing, there was a huge problem at first. The high heat you need for the reaction would destroy the builder enzyme every single time. Scientists literally had to stand there and add more of it by hand every single cycle. It was tedious. The real game changer came from a pretty unlikely place, the boiling hot springs of Yellowstone National Park. Scientists found Taq polymerase, an enzyme from a heat-loving bacteria that actually thrives in that environment. This little microbe's ability to survive the heat was the key that finally automated the whole process and turned PCR into the powerhouse it is today. Okay, so we've got our ingredients all mixed together. Now for the actual process. See, the real magic of PCR isn't just in the mix. It's in a repeated cycle of very specific, very rapid temperature changes. This whole shebang happens inside a machine called a thermal cycler. Just think of it as a super precise little oven that can heat up and cool down incredibly fast. It just follows a simple program, and each one of those peaks and valleys in temperature that you see on this graph, well, each one triggers a critical step in the DNA copying process. And it all boils down to this simple three-step dance that just repeats. Step 1. Denaturation. The machine cranks the heat way up to almost boiling. This forces the DNA's double helix to unzip into 2 single strands. Step 2, annealing. It cools right down, which allows our little primer bookmarks to find and stick to their exact target sequence. And finally, step 3, extension. The temperature comes back up to the perfect sweet spot for our Taq polymerase. It locks onto those primers and just starts building, racing down the DNA strand, creating a brand new copy. And boom, that's one cycle done. As you can see right here, we started with one piece of DNA, and now we have 2. The real magic, though, is that we can just repeat this simple process over and over and over again. Now, this is where it gets truly mind-blowing. You don't just do this once. The machine immediately starts the next cycle. So after 2 cycles, you have 4 copies, then 8, then 16. It's growing exponentially. After just 30 or so cycles, which only takes a couple of hours, that one single starting molecule of DNA has been amplified into over 1 billion copies. Just incredible. So that's the classic PCR recipe. But you know scientists, they're always tinkering, always innovating. They've built on this core idea and created a whole family of specialized PCR techniques. And of course, each one has its own special acronym that can get a little confusing. Yeah, the number of different types of PCR can be a little intimidating. So let's just quickly clear up 2 of the most common and most important ones you're likely to hear about, RT-PCR and qPCR. Okay, so on one side you have RT-PCR.
The RT here stands for reverse transcription. The key thing to know is that this technique actually starts with RNA, say from a virus, and it uses a special enzyme to turn that RNA into DNA before the regular copying process even begins. So it's basically answering the question, is this specific bit of RNA present in my sample? Then on the other side, you've got qPCR, or quantitative PCR. This one's different. It cleverly uses fluorescent dyes to measure how much DNA is being made in real time. So instead of just yes or no, it answers the question, okay, but how much of this DNA is in my sample?
All right, so we've seen how this molecular photocopier works, but why does it matter so much? The answer is that this single technique has woven its way into almost every corner of modern science and medicine. It's everywhere, from solving crimes to diagnosing diseases. In medicine, PCR is an absolute diagnostic powerhouse. It is the technology behind the tests for countless infectious diseases, from HIV to the flu and, as we all know now, COVID-19. It's also used to detect genetic disorders, to help personalize cancer treatments by looking for specific mutations, and even to do the crucial tissue matching that's needed for organ transplants. And of course, there's forensics. If you have ever watched a crime drama, you have seen PCR in action. It's the engine that drives DNA fingerprinting, letting investigators create a genetic profile from the tiniest speck of evidence left behind. It's also the gold standard for paternity tests and for identifying human remains after a tragedy. But its reach goes so much further. PCR was one of the key technologies that made mapping the entire human genome even possible. It lets scientists pull out and study ancient DNA from fossils of long-extinct animals. And in industry, it's used every day to test our food for harmful bacteria or to detect GMOs. Honestly, its impact across all of science is almost impossible to overstate. This quote really just says it all, doesn't it? The invention of PCR wasn't just another small step forward. It is widely considered a genuine turning point, one of the most important scientific advances in the history of biology, a total and complete game changer. So when you really boil it all down, it's such a beautifully simple idea, right? Just a cycle of heating and cooling that lets a tiny enzyme do its job. But that incredibly simple process gave us the power to read the story of life itself with a clarity we never had before. It has already changed our world in countless ways, which just makes you wonder, what will we discover with it next?